Hello students, we are now going to look at lecture 7 of Introduction to Food Chemistry, and we want to understand the sugar chemistry in food. Sweetness levels vary among different sugars, and also crystallization patterns can influence the texture of various sugars. In one of the classes, we looked at two types of reactions: the enzymatic browning, the non-enzymatic browning, and we also looked at the Maillard reaction. The Maillard reaction occurs between sugar carbonyls and amino acids, and it produces browning and flavor compounds. Caramelization is also one of the processes that occurs at high temperatures, and here you find that sugars decompose, forming brown compounds, which results in flavor, color, and also loss of nutrients. Reducing sugars. A sugar can be called a reducing sugar if it has a free aldehyde group. Ketoses can act as reducing sugars only after isomerizing to an aldose. The aldehyde carbonyl group acts as a reductant, hence the term reducing sugar. Sucrose. Sucrose is formed from glucose and fructose, and it is a non-reducing sugar. Both anomeric forms or both anomeric carbons form a glycosidic bond, and the ring cannot open. It has a closed ring configuration. It can be hydrolyzed by invertase enzyme into glucose and fructose. It can also be used in baking, in sweets, and golden syrup production. Galactose and trisaccharides in beans. The main oligosaccharides in beans include raffinose, starchose, and verbascose. They contain galactose, fructose, and glucose units. The human gut cannot digest enzymes, does not have the enzymes to digest these forms of carbohydrates. And so they are fermented by the gut bacteria. This is the fiber that we usually talk about. Fiber cannot be broken down by the enzymes in the digestive tract, but it can be fermented by the gut microbiota or the gut bacteria. And this leads to production of some chemical products such as methane and carbon dioxide. And this causes flatulence in the body. This is very typical of beans. Usually when a person consumes beans, within an hour or two of consumption, once digestion has taken place and the fiber goes down into the colon, because it cannot be broken down in the small intestine, but can be fermented in the colon, that is the large intestine, by the gut bacteria. Once this fermentation takes place, carbon dioxide and methane gas is produced, and this causes bloating in the body. And the end product is, you find that someone is continuously gassing or passing gas or polluting. And this kind of gas is usually very smelly, owing to the presence of these gases. The structural examples of trisaccharides, we have raffinose, which constitutes galactose, glucose, and fructose. Starchose constitutes galactose, and then glucose and fructose. Verbascose constitutes 3 galactose units, glucose, and fructose. So you can see that the chain and the naming of each of these. You find that raffinose contains 1 galactose, starchose contains 2 galactoses, and then verbascose contains 2 galactose units, and then you add on glucose and fructose. And all these involve the alpha 1,6 and alpha 1,2 glycosidic bonds. Polysaccharides. High molecular weight carbohydrates are made of many monosaccharides. And here are the examples. We have starch that provide energy storage in plants, cellulose, important for structure of plants, and glycogen, energy storage in animals. Physical properties of sugars. Sugars are typically white and crystalline or amorphous. Sucrose is crystalline and glucose is amorphous. And it's highly water soluble. For example, 2 kilograms of sucrose in water. Chiral, they can rotate polarized light or they are optically active. Non-enzymatic browning, and this is the process we call caramelization. Sugars can decompose at a temperature of 160 to 180 degrees Celsius before melting, and they release water, leading to browning. And the products here we have hydroxy, methyl furfural, then acrolein and pyrovaldehyde, and then we also have glyoxal. And some of these caramels contribute to caramel flavor and produce burnt taste and smell. The Maillard reaction, as we noted already, is the reaction between sugars and proteins or the amino acids, and it produces flavors and aromas in foods such as bread, cookies, and nuts. Advanced Maillard products can also smell or taste burnt, and these may be toxic to the body. Forms compounds like pyrazines. Carbohydrate analysis. Carbohydrates, we said, contain carbon, hydrogen, and oxygen, but they are structurally complex. In food analysis, total carbohydrates is equal to, it can be calculated by difference, and it can be measured after moisture, fat, protein, and ash. Analytical techniques used for sugars. We can use the UV-Vis spectrophotometry, and this only works after pretreatment to form absorbing compounds. For example, the glucose test strips. We have also the IR spectroscopy. This one has limited use. You find that water strongly absorbs the IR. Then we have the HPLC-MS, which is best for analyzing complex sugar mixtures. Polarimetry is useful for pure sugars and uses optical rotation. And then iodine starch test. Here you find that iodine plus iodide plus starch produces a blue complex. Then gas chromatography is not very useful, and here I find that sugars decompose and can damage the columns. That's why it's not a very reliable method of sugar analysis. Thank you and have a good time.